And after the class, as you will find out, I do put up the videos on YouTube, and they'll be available throughout the semester. So here is my information. I am Dr. Paula Waziri, and here is my email. And I will hold office hours, again, temporarily on Zoom until I can physically get back to the US, which is going to be hopefully by September 4 or 5. In the meantime, I am holding Zoom office hours. And when? That's going to be on Thursdays and Fridays. And here is the time that I'll be holding the office hours. If you cannot make those Zoom office hours, make sure to drop me an email, get in touch with me, and I will get back to you, and we'll set up some other time outside of these office hours. And our TA is Belle, and she should be in the class right now, like saying hi. Say hi, Belle. I assume she said hi. And her office hours are going to be right after the class on Wednesday, and we're planning to use the same room as we're using now, and hopefully everybody found the correct room without going to the old room, and everybody's in class right now. So anyway, we'll see if the class is not in use afterwards. Then Belle is just going to hang around and hold her office hours on Wednesdays. Anyway, here is her information. And I believe this class also is set up for supplemental instruction for the SI. But the SI leader did not contact me yet, so I'm assuming that by next week the person is going to get in contact with us. So the textbook that we use is Molecular Biology of the Cell. And I know there was a bit of a discrepancy in the simple syllabus because there were 2 editions there, and I got some emails saying that the bookstore had the 6th edition. And basically those editions are about the same. There is not really a lot of difference between the 2 editions. So if you have the digital edition, the 7th edition, that is fine. If you prefer to carry that brick on your backpack and you want to buy the physical book and the bookstore has a 6th edition, that is fine with me too. And the reason is that I mainly base the material for the class in the PowerPoints. So with any of the editions, if you just go by the PowerPoint topics and you look for those topics in whatever edition you have, you're going to find the same information. So I'm happy with any of those editions. I'm happy with the PDF files of the book, and whatever is more practical and affordable. So here, how are you going to pass this class? What do you need to do to get your grades? I'm going to use scores from 4 exams. Okay, so 4 exams will give you 100% of your grade. Of course, that gives you 25% total per exam, right? However, you will have available a 5th exam, which is the cumulative final exam. Okay, so the cumulative final exam is going to be given at the same, on the same day as your 4th unit exam. And the cumulative final is not mandatory, but you can take the cumulative final if you just love the class so much that you just want to see what is in the exam, or if you did not do so well in one of the 4 unit exams and you want to drop one of your lowest grades. Okay, so if you take your cumulative final and you do well on the final, then you drop the lowest grade. If you take the cumulative final and you do very badly on it, then that is your lowest grade and it gets dropped. Now, don't panic when you look at the Canvas and you see that there will be a 0 for the cumulative final if you did not take the exam, because that's the grade that's going to be dropped. I have a certain set of rules that I need to input in Canvas in order for it to work. Another thing, make sure that you calculate your grade, because Canvas sometimes messes up the grades, like more times than I would expect it to. All right, so here are 100% of your points. Besides those points, you also have the in-class extra credit grade. Now, that is going to happen through the iClicker. As if you have read the syllabus, the simple syllabus, you'll see that we do use iClicker in the class. Now, I prefer if you use the app version on your phone, because we will be in general participating on a cool research that the Macmillan company is doing on the iClicker usage, and you will be contacted, by the iClicker team, soon, in order to, either opt in or opt out. Is your preference if you want to participate on it or not. But I really see that the people who do show up in person in class and take advantage of these extra credits, they end up doing a lot better in the class than everybody else that decides to not show up to class. Okay, so attendance is not mandatory, but if you show up, you get points for it, so why not, right? And as you can imagine, the class is not that easy. I really try to make it as easy as I can, but the subject itself, it's a upper class type of thing, so it gets complicated sometimes. All right, so I was asked to add this extra information on my first day of class, and this has to do with a scholarship for students. I believe that might not be very relevant to this class, because they say you should be starting college or in your earlier years, but take a look at it. If it applies to you and you want to apply to the scholarship, go for it. Why not? Okay. All right, so the division of the class. So we have 4 units. The first unit is including here the 3 first chapters. So the first units are going to be for weeks 1, 2, and 3, and those topics are going to show up in your unit exam 1. And when is that going to be? September 10. So hopefully I will be back in the class, in person, back in the US, and not fighting with the CDC any longer, hopefully. Okay. So that's your unit 1. Unit 2, here are the chapters, okay? So here is going to be those topics are going to be in your unit exam 2. That's going to be October 10. And here you have your unit 3, where we're still going to, get not as many topics as in unit 2, but again, individual topics, they're going to be on exam 3. And this is all in the syllabus, and you can follow along which topics we are studying for each of the units. And I have added here information that I just looked up about the day for your unit exam 4 and your final cumulative exam. Okay, so unit exam 4 and cumulative exam 5. That's going to be on December 10, is a Wednesday, and the time is different than our meeting time. All right. Okay, so objective of the course. What is it that you are going to be looking at? Basically, we're going to look at how cells are functioning. So we're going to look at molecular machines, and we're also going to take a good look at how enzymes are functioning. Okay, we're going to look at the structure of enzymes and particular ways that allow enzymes to work, and also going to see how this entire life form of cells allow it to survive. Okay, so we're going to look at tiny enzymes and bigger machines, and then we're going to try to relate one system to another, and try to figure out what is it that is making the cell to maintain life. Okay, so we will be focusing on how these structures work and how is it that they are related in case of their functions in different species, because several of these machines are really evolutionarily conserved. So we're going to briefly take a look on how this type of function is carried on throughout the species. But again, we're not really going to dive a lot into the genetics of the molecular genetical evolution, because this is not a genetics class. It is a cell biology class. Okay. So here are some strategies for learning. Again, of course, everybody learns at different paces, and also you might know by now what is the best method that you use for your own understanding of the topics, right? But basic things that you should do. You should review some biochemistry, and you should definitely review some energetics, right? Some molecular behaviors and energies of certain molecules in comparison to others, and energy of some chemical bonds, right? But this is not biochemistry class. So whatever you need for this basic biochemistry knowledge, I will be presenting in the class. So you focus on those sections that I'll give you throughout the PowerPoints. Okay. Again, we're going to really be looking at fundamental processes of machineries, of these tiny machineries of the cells, and again, how they are related to the overall cell function. Okay. Again, you should be looking at why certain thing is working, why certain machines are powered by the energy demands or the availability of energy in the cells. So you should always be thinking, why is this important for cellular function? If this part doesn't work well, why is it going to be detrimental? What are the consequences if certain machinery is not working properly? All right, so what is it that you should do? You should go over the PowerPoints, like before coming to class. So it's not a surprise when you come to class and like, oh, she's talking about this or about that. So take a look at the PowerPoints, right? And if you can, go through the textbook, right? Open the textbook and try to understand what the textbook is telling you. And I know it's a huge book. The chapters are huge, enormous, and the language sometimes gets to be a bit confusing. And if you see that you are reading a chapter and you're falling asleep on it, that's not very effective reading or learning. So make sure that when you're spending your time reading the textbook, don't panic and focus your attention to what is it that the book is trying to tell you. All right? Do this bit of exercise before.
Dr. Waziri,
yeah?
I'm sorry to interrupt. I thought I would be really clever and bring the IT person in while you were talking to fix stuff, but we've run into a tiny issue. I'm going to need to close and restart your Zoom meeting.
Okay, I shall pause the recording.
How do you like me so far?
All right, here we go. All right, so there is no such thing as a bad question. So please ask questions. And even with this, strange setting here online, we'll still be able to ask questions and communicate. All right, so here, just to reiterate how you're going to be assessed in the class, you have the 4 unit exams, and those have to be multiple choice questions, right? So each exam is worth 100 points. You have an average 75 minutes for each of them. And I usually ask multiple choice. Rarely I put in some true and false, but mainly multiple choice. Okay. And here again, you do have your extra credit for iClicker. How much of this counts? You can have up to 10% of extra grade in your class. So let's say that you have 90 overall average in 4 exams, and if you have 100% of the extra credits earned, then that will push your grade to 100. So simple math here. So here is the code that you should use to join the iClicker, and I did add that to the PDF file, so you should have that handy. Okay, Okay, again, just I prefer for you to use the app, but if you have the remote, then go ahead and use the remote. Now, how are you going to be graded for extra credit? Showing up in class and turning on your app, opening your app or turning on the remote, you already get 1 point. So that is your participation for showing up type of point. Okay. When you try to answer a question, even if you answer it wrong, you also get a point. So even if you're asleep and I click or question shows up, you're like, oh, I will just answer A. So you get a point. If you answer it correctly, then you get 2 points for that question. Okay, now, again, do not vote for people who are not present in the class. That is considered cheating. If you miss one exam and you have a good accepted excuse, then we'll work for you to make up that exam. If you miss a class because you cannot show up and you wanted to recover your iClicker points, forget it. There are no makeups for iClicker, okay? If you miss an exam and you have a good excuse and documentation to prove your good excuse, then we will work with you to make up the exam. Okay. If you miss an exam for any reason, including one excuse that is not accepted, then plan on taking the cumulative final because you do need grades from 4 exams. How are you going to do well in this course? You're going to focus on the topics that I add in the PowerPoints. And you probably have heard that I usually base my questions on what I teach in the PowerPoints and what I teach in the class. Attend every lecture and make sure that you are paying attention. Make sure that you are awake. Keep up with the topics, right? Read and study and understand what is written on those topics in the textbook, okay? Go to those SI sessions. I still don't know who is coordinating them or when they're going to be, but yes, I'm pretty sure there'll be SI sessions for this class. And yes, use artificial intelligence, but be careful because some of the feedback from this AI is not very accurate. How is it that you're going to use the AI? Well, I will have more examples as we go along. But basically, you can, let's say, if you're going over one slide in the PowerPoint and you want to make sure that you really understand and you're thinking what kind of questions I can ask about this certain topic, go open your AI tool. Like I like to use, ChatGPT, and ChatGPT 5 just came out. But you have the free versions as well. Open your AI tool and say, give me multiple choice questions based on this topic. Now, if the AI tool gives you certain questions or certain answers that do not make a lot of sense, don't believe the AI tool right away, okay? Make sure you double check with the textbook, double check with the PowerPoint, and ask me or ask the TA, okay? Because sometimes, more often than I would like, AI has some crazy ideas and weird explanations, all right? So use it to reinforce what you already are learning, but do not rely on it too much. Okay, so again, ask questions and visit us in office hours or virtually. Make sure that you keep up with the course. And now finally, we get to chapter 1, and I do hope that we can cover the whole thing because I like the last slides of this PowerPoint. So here we go, chapter 1. What we're going to study here, genomes and cells and diversity of life. And I believe this is when Dr. Thiessen is going to pick up.
But it's there to simply support, take up matter, take up energy, make a copy of yourself, repeat. That's it. Nothing less than a cell can do that. And you don't have to be more than a cell in order to be able to do that. That's why we study cells. That's where the rubber hits the road. That's where non-living molecules. So to accomplish this, cells, they synthesize and they use a couple of basic structures: molecular switches, molecular machines, molecular catalysts, which we refer to as enzymes, molecular structures, holding these things together. And that's what's going to essentially make up the core of this course, is looking at those different structures. So this is an iClicker question. So the question, okay, so go ahead and read the question. Which of the following best describes why cells are studied? They are the smallest non-living unit of matter. They are the basic living unit of life. They are composed entirely of proteins, or they cannot reproduce on their own. So does everybody know how to do their frequency? There's little instructions on the back. What's amazing is that of all of the life forms on this planet, there are a surprising number of things that you can say that apply to all of them, right? There are over 2 million described species on this planet, and almost certainly a whole bunch more than that that have yet to be described. Each of those different species has its own set of hereditary information that defines the organism itself and all of the machinery necessary to reproduce, right? It's the hereditary information in the fertilized egg cell determines the nature of the whole multicellular organism that will develop from it. Again, one thing to keep in mind, I'm sure most of you already know this, but it's quite remarkable to recognize that the DNA in this egg cell is identical to the DNA in this adult urchin. The DNA in this egg cell above the rabbit, or the mouse, I should say, is identical to the DNA in the adult mouse, right? And that's true for every single organism. So one of the challenges in studying cell biology, first, is to appreciate how tiny things are relative to the world in which we live. It's a challenge to just think of that conceptually. If you're a researcher, it's a whole nother challenge to actually try and observe, study, and understand things that are so, so tiny, right? So atoms are the basis of all molecules. All molecules in the cell, therefore, are made of atoms. Atoms are extremely small, very difficult to study. Fortunately, as biologists, we're able to rely heavily on chemists and physicists to tell us about the properties of atoms. This is what they call a buckyball, this C60, this carbon sphere made up of 60 carbon atoms, a rather unusual structure, about a nanometer in size, smaller than the average lipid or the average protein, which is maybe 5 to 10 nanometers in size. A flu virus, you're getting a little bit bigger, 100 nanometers, but as I'm sure you can imagine, a virus, which many, many, many thousands of copies could fit inside of a eukaryotic cell, is many times smaller than a typical cell. Mitochondria in bacteria are approximately the same size in most cases. You may know that mitochondria are supported by evidence to have evolved as free-living symbiotic bacteria. Red blood cells are very small in the terms of eukaryotic cells. Animal and plant cells, eukaryotic cells a bit larger. Pollen is a reproductive cell for many plants, a single cell. Human egg, also a single cell. A frog egg is a very large egg cell, still a single cell. That's why you may know that frogs and frog cells and frog embryos are often the targets for different types of cell biological studies, because they are a bit larger and therefore a little bit easier to see than other types of cells. So most species on this planet are prokaryotic. In fact, most is almost an understatement. It would be probably more accurate to say almost all cells are prokaryotic, and every single prokaryotic cell is unicellular, all right? So prokaryotic literally means as old as life. There are two subcategories within the prokaryotic group, the eubacteria and the archaean organisms. They have what we might call a simple genome. Simple is in quotes because we need to understand in biology, we refer to things as being simple or complex. Complex does not mean complicated. It means more. So when we say simple genome, what we mean is it has fewer protein coding regions typically than a eukaryotic organism would have. That doesn't mean it's not complicated. Many prokaryotic organisms live in extremely challenging environments that we wouldn't have even the remotest chance of being able to figure out. They have a single circular DNA molecule in almost every case. There are very, very few exceptions to that, but by and large, all have a single circular DNA molecule. It resides in the cytosol. It's not separated from the rest of the cytosol by any kind of a membrane-bound compartment, as we see in eukaryotic organisms. Again, all are unicellular. Now, there are colonial forms. Lots of bacteria cooperate to form colonies. The difference is that in that case, each of those cells is its own individual reproductive unit, and they are essentially identical to each other, whatever minor mutations they might have between them notwithstanding. So they are colonial as a mechanism of cooperation, but they're still very much in competition with each other for reproduction, right? Most prokaryotic organisms have a cell wall. None of them have membrane enclosed organelles, what we might call true organelles, and they're extremely diverse. If there is even the remotest possibility of scrapping up enough energy and enough matter to make a copy of yourself, no matter how harsh the conditions might be by our measure, a prokaryotic organism has figured out how to do it. Now, it's a little bit misleading when you say there's been about 20,000 species of bacteria named, because the overwhelming majority of bacterial species have not really been encountered, much less named. So there's probably well over 10 or even 20 or even billion species of prokaryotic organisms. It's really almost impossible to say how many. It's certainly far more than 20,000, but that's how many have been formally named. Beg your pardon? Far more than insects. Even though prokaryotic organisms do not have membrane-bound internal compartments, what we might call true organelles, that doesn't mean they're not capable of creating unique microenvironments in which concentrations of specific types of molecules can interact with each other to carry out specific types of chemical reactions. There are a couple of different types of internal organization, what we call carboxysomes, which are protein encapsulated structures, nanocompartments, and microcompartments. These are both protein-coated compartments, but without that lipid bilayer that defines a true biological membrane. And then a really interesting kind of structure called a phase-separated compartment. This is a little bit difficult to understand, explain, appreciate if you're not a physicist, but it basically, in a really simple way, if you take oil and you drop it onto a bucket of water, the oil kind of coalesces in groups with itself. The water stays together. That's a phase separation. You can mix it all you want, and it will still eventually separate out. The thing about the bacterial organism is they can control that phase separation. So sometimes the oil and the water are actually really nicely mixed. And in other cases, because of changes in things like pH and other factors, they will create this phase separation. And within these different phases, they carry out different types of chemical reactions. The benefit of these compartments in bacteria, the same as the benefit of the compartment in a eukaryotic organism. It allows you to create little micro laboratories within the larger laboratory to carry out really specific chemical reactions without having to worry about the other chemicals that might interfere with the chemical reaction you're carrying out in this location. Now, having said that, yes, go ahead. Okay, got it? All right. So, having said that most organisms are prokaryotic, we obviously know that some are eukaryotic. It's very hard for us to appreciate that there are almost no eukaryotic cells compared to prokaryotic. Our entire world is essentially made up of interaction with eukaryotic organisms, right? But there are still far fewer of them. We're understandably a bit more interested in them in many cases, but as hopefully Dr. Waziri will get to at the end of today's lecture, we probably should be far more interested in prokaryotic organisms than we actually are. So eukaryotic means evolutionarily young. They are characterized by having linear DNA molecules, and with very, very few exceptions, they have more than one DNA molecule. Some have very few. Drosophila has 6. A few ant species have fewer. A moth has over 400. So there is a wide range of the number of DNA molecules. Now, the number of protein coding regions is not horribly variable. It's just a matter of how big or how small the DNA molecules are. So the moth, obviously, with 450 DNA molecules, those are each very, very small. But probably one of the more significant features of the eukaryotic DNA is that it's separated from the rest of the cell within its own membrane enclosed compartment, what we call the nucleus. This has actually led to lots and lots of other differences, and without going into tremendous detail, one of the things that has happened since these cells evolved the nucleus and separated the nucleus from the rest of the cell is the DNA was able to get larger through environmental acquisition, viral infection, other mechanisms that add DNA to an organism's genome. It was allowed to get bigger because as it got bigger, it was separated and didn't interfere with other chemical reactions. We talk about sometimes the benefit of separating spaces within the cell to create these little micro laboratories. Well, things actually can bump into each other and interfere with each other if they get to be too big, too cumbersome. So in many cases, in the case of prokaryotic organisms, DNA can't really get much bigger than it is right now because it will begin to interfere with cell processes. But once inside the nucleus, it could get bigger. Bigger DNA means more potential coding regions. It means the cell can get bigger. It actually also means it's harder to move things around. So a cytoskeleton evolved in order to move things around. That meant the cell wall was no longer needed. The cell wall was lost. That allowed phagocytosis. Bacteria were taken into phagocytosis and somehow survived the ordeal and went on to become mitochondria and chloroplasts. Lots of the DNA is not coding, it's regulatory. That means we can control very, 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 very intricately what we do and do not turn into protein from our DNA. That allows us to be multicellular. The reason prokaryotic organisms are not multicellular, they don't have a big enough genome and they can't pick and choose different genes to express in different types of cells. All right? Only plants have a cell wall. The cell wall we find in eukaryotic plants was evolved later when we lost our cell wall because we didn't need it anymore. We were still living in an aqueous environment. As we began to move onto land, some of the cells began to assemble themselves upward against the force of gravity. Cell wall became useful again. So these organisms evolved an entirely new cell wall, completely different, evolutionarily unrelated to the cell wall we see in prokaryotic organisms. So when we talk about cell wall, we're talking about basically a polysaccharide-derived structure that is made originally in the ER, goes through the Golgi. There are numbers of different kinds of cells that have different kinds of rigid support structures, if you will, but you know, we don't want to we can subcategorize a bit. We say animal cells do not have a cell wall, but some animal cells do build some types of, you know, kind of structural support systems, not really a true cell wall. Less diversity in the prokaryotic organisms. We actually have a approximate count of eukaryotic organisms, about 8 to 9 million species. Is what the estimate is. Maybe a third of those, a quarter of those have actually been described, but we have a little bit more of a handle on what's likely to be out there in the case of eukaryotic organisms. So most eukaryotic organisms are unicellular. A lot of us are probably familiar with some of these, looking through a microscope at some point in the past, seeing the paramecium run around, seeing the amoeba do stuff. So again, most cells are prokaryotic. Of the few that are eukaryotic, most of them are single cell. So the overwhelming majority of cells on this planet are single cell, all right? So this is a picture of a...
This single cell amoeba grows around by using actin polymerization. Growing actin filaments push out pseudopods, or falsely, allowing the cell to explore new territory. At the same time, organelles move along cytoskeletal tracks within the cell.
So what you can't see in that image is that underneath that movement or supporting that movement is a network of protein filaments that these things are all traveling along. Now, I don't want the single celled amoeba to get all of the attention. Cells in our bodies crawl around too. This is a white blood cell chasing a Staphylococcus aureus. And you root for the bacteria or the white blood cell? White blood cell. Wait. Got it cornered almost. It's like Pac-Man. Got it. So our cells move around as well, and they do so by a variety of mechanisms. This white blood cell happens to do it by a mechanism that is very, very similar to the mechanism by which the amoebas move. I beg your pardon? That's a good question. No. I would say no. All right. So even though all prokaryotics are single cell, most eukaryotic organisms are single cell, some eukaryotic organisms are multicell, as we obviously know. What's interesting about a multicellular organism, out of a single type of DNA, they can make different kinds of cells. And they do this because, let's say they need 30 to 50,000 proteins to be whatever it is they are, but they've got 100,000 proteins to choose from. They simply pick a different combination of proteins in a liver cell versus a skin cell. In plants and fungi, you have approximately 10 to 20 different types of cells, again, all made from the exact same genome. In mammals, you have somewhere between 1 to 200 different types of cells. Humans, about 200 different types of cells, again, all made from the same genome, obviously all descended from the very same single fertilized cell. So if we compare just kind of side by side quickly prokaryotic to eukaryotic, all prokaryotic organisms are unicellular, have a single circular DNA molecule that resides within the cytosol, not separated. Eukaryotic organism, we have both single and multi. They have multiple DNA molecules. Those DNA molecules are linear. They are housed within a separate space, a nucleus, right? By comparison, a prokaryotic organism has a simpler genome, and an eukaryotic organism has a more complex genome. Now, when I say complex, I mean more of something. More of what? More protein coding regions is generally what we are referring to. We could also say more DNA, but we don't mean more complex. Which organism synthesizes more different kinds of proteins, a human or a carrot? A carrot. So a carrot, by this definition, is more complex than a human, all right? Plants generally do have more protein populations, larger protein populations, probably because it's a little harder to maintain homeostasis when you can't move to a better environment. No organelles in prokaryotic organisms, but of course, remember, they still have the ability to create these little micro laboratories, which are very important. Eukaryotic organisms take that benefit to the next level by having membrane enclosed internal compartments. All prokaryotic organisms have a cell wall, with a few exceptions. In eukaryotic organisms, we find a true cell wall only in plants. Prokaryotic organisms evolutionarily ancient. Eukaryotic organisms evolutionarily very, very, very young. One could easily argue that whether or not eukaryotic organisms are a successful strategy is far from answered at this point in our evolutionary history. Huge number of species in prokaryotic organisms, relatively few in eukaryotic, a tremendous difference in diversity, and prokaryotic organisms make up well over 99% of all cells on the planet, right? All right, and in humans, there are more than likely more prokaryotic cells living than there are your own eukaryotic cells. Cells that you could not survive without, as it turns out. And we're learning more and more about that pretty much every day. So with that, I'm going to ask you an eye clicker and then turn it back over to Dr. Waziri to finish up. And we do actually have a few minutes. Does it go to 3:20 or 3:30? 3:20. So she may want to, I don't know, I'll let her decide. So which of the following is a defining feature of a eukaryotic cell? That it has circular DNA free in the cytoplasm, all are unicellular only, all possess a rigid cell wall, or presence of membrane-bound organelles. Which of these defines a eukaryotic organism? So again, once I push the start button, you have about, again, wait a second or two before you vote. You'll have about 30 seconds to cast your vote. Yes. So there we go. All right. So with that, I am going to turn it back over to, I think, Dr. Waziri at this point. Yes? All right. So I am going to stop sharing, which should... Okay, Dr. Waziri, we have about 5 minutes.
All right, so here I'll go for the Speedy Gonzalez talk on the very interesting part on the 5 minutes of the class. All right, so at first, when you look at the DNA content in bacteria as well as archaea, we see that the bigger the DNA molecule gets, that goes hand in hand with how complex that certain organism gets. Okay. Now, when we get to eukaryotes, then we're not going to find that linear relationship. So, for example, here, when you look at the DNA of bacteria or protists here, we see that they have a lot of DNA, and then we go down here just going to see mammals. Look, it's not a lot of DNA. Okay, so that is kind of complicated because we're thinking, well, these are very, very complex or multicellular organisms, and how come they don't have a lot of DNA in their nuclei? So this discrepancy is actually what is called this C-value paradox. Now, what that means is that the amount of genes that are expressed they don't really go hand in hand with the amount of genes that are coded in the DNA cell. In this case, it says the haploid genome, because as you know, we have diploid genomes, being that you get one chromosome from your mom and another one from your dad. So if you look at all the genes of the haploid genome, you see that it's not a lot of genes in comparison to those more primitive types of organisms. Now, interestingly, we know that the Genome Project that was meant to sequence all of the genes in the human genome, at first, the Genome Project was sequencing only gene or protein encoding genes. And at first, the scientists found about 20,000 genes in the human genome. Look at this, it's only a little bit. But we know that in average, we need about, I don't know, about 100,000 genes in order for our bodies to function. And the question was, where are all the other genes coming from? How are we getting to express all the other genes if they are not coded by our own gene and protein encoding genome? So then there was a second phase of the Human Genome Project that just finished in 2022. And in this sequencing strategy, the group was really looking at not only protein encoding genes, but they're also looking at non-coding genes and pseudogenes type of models. So if you put everything, including the coding genes, in this sequencing strategy, then we found about nearly 64,000 genes. Still, we need a lot more genes being expressed in the body. And the reality is that the rest of the genes that we need to be expressed and functioning in our bodies, they're not coming from our genome. They're not coming from our cells. They really are coming from the microbiome that lives in us and on us. So if you look at the sequencing of microbiome that we know so far, they will have about 3.3 million genes that are expressed on us and in us, and that is really a lot more than the amount of genes that we have in our chromosomes. Now, I know that we're going up the time. At this time, the class should be over. So I'm not going to just rush through these next slides, but I will start the next class on Friday. I will start them with these slides because these I find to be very, very interesting and should be mind-boggling for you. So I'm going to stop here. And